focusing on finding pharmacokinetic data. My name is Anne-Marie, and I'm your host today. I'm here with our PharmaPendium expert, Pooja Jain. Pooja comes with over 10 years of experience in the life sciences industry, where she's experienced working, for example, at Mimetogen Pharmaceuticals and Eli Lilly. Pooja has both a master's in molecular biology and an MBA degree, and is here to educate us more on the pharmacokinetic module of PharmaPendium, such as how you may make drug development project risk assessments, identify competitive opportunities in the therapeutic area or drug class, and gain critical understanding to potential pharmacokinetic responses by looking at critical data such as food effects, concomitant drugs, comorbidities, and demographic effects. We will be placing this webinar online so you can listen to it again at your own pace. This webinar is aimed at users with some experience with PharmaPendium. However, we do also have introduction webinars to PharmaPendium, and the next introductory webinar is scheduled for June 27th. We'll remind you of this at the end of this webinar as well. Before we do get started, I would briefly like to put PharmaPendium into context with the other activities that go on at Elsevier. Elsevier serves three main and interlinked areas which may be of interest to you: biology, chemistry, and medicine. PharmaPendium gives you searchable access to all FDA and EMEA approval documentation, and we also provide additional solutions serving the biology and medicine spheres, such as Embase for your biomedical literature searching needs, Cyverse Science Direct for full text access, and Cyverse Scopus, an all science abstract and citation database. This should help to put PharmaPendium into context, and with that we are almost ready to move to today's session with Pooja. But just before we get started, a couple of need-to-knows. During the session, you may send us your questions by using the chat or ask a question function on the webinar control panel. We'll have 15 minutes for questions or thereabouts at the end. And for all questions that we do not answer in this webinar because of time, we'll include in a complete Q&A and send to all of you after this webinar. If you wish to have a full screen view, please click on the red arrow. Your control panel will be hidden. Click again on the red arrow to see your control panel again. And with that, I would like to welcome Pooja. And Pooja, can you tell us all about how to search for pharmacokinetic data in PharmaPendium?
Sure, and thank you, Anne-Marie. Welcome everyone to the Pharmacokinetics module webinar. So what we're going to do right now is start off with an introduction as to what the Pharmacokinetic module is. From there we'll go into a demo. We'll start off then with a poll question after a brief demo, and then do more of a deep dive into some interesting examples. So let's start off with the Pharmacokinetics module. That module is basically giving you unprecedented access to preclinical and clinical exposure data extracted from FDA packages, both current and historic documents, and European documents. We have at least 2,000 drugs with preclinical and clinical exposure data at multiple parameters and usually under various experimental conditions. So you can actually get very interesting data on concomitant drugs, disease states, demographic differences, etc. Now what's interesting is that the level of PK data to be contained in this database is almost never published, a lot of times for competitive reasons. And we have extracted a lot of this competitive data and put it together in a very user-friendly format. We have the Pharmacokinetics preclinical and clinical and drug safety databases browsable within the same platform, so you can get a lot of data all within one easy viewable browser. We have extracted parameter values for at least 2,000 drugs. We have taken this from FDA documents stemming back from 1938 all the way to the present time. And a lot of these documents are present in the FDA Classic Collection, which basically has FDA documents from 1938 to 1991 exclusive. Now, if you do not have access to the FDA Classic Collection, that is a separate purchase from the PharmaPendium platform, you will have access to the extracted data and citation, but you will not be able to drill into the original document. Sometimes drilling into the original documents is actually quite useful because you can gain access to a lot of other information, which you will see as we go into the demo. we also have the European regulatory documents from 1995 to present. In terms of the amount of data which we have extracted, we have an estimated 1.25 million lines of extracted pharmacokinetic data, with filterable fields and special designations for things like drug names, species, study groups, enantiomers, metabolites, tissue-specific studies. You can get a lot of data right at the tip of your fingers within a few minutes, as opposed to spending weeks or even months trying to get this data. Now, what are some reasons why you would use the pharmacokinetic module? Well, for one, you can create modeling sets so you can have valuable data in modeling, for example, therapeutic windows for new drugs. You can use it for drug development project assessments, so you can have a newly available wealth of comparative data to aid in development of candidate decisions, prioritizations, and direction. And you can also use it for competitive analysis, so you can actually ask the questions, can we deliver this drug better than our competitors? Where has the bar been set in this drug class? And where are the opportunities in drug class X, for example, using our proprietary methods and formulations? So for example, this can be really interesting when you're looking at drug repurposing studies. Getting access to a lot of comparative data can really speed up the process for getting approval for drugs that are being approved through the 505(b)(2) pathway, for example, with the FDA. So with that said, let's now get into the exciting part of the webinar and go into a few demos. So here you will see the home page of PharmaPendium. And now to get into the pharmacokinetic module, what I'm going to do here is click on pharmacokinetic data. And once I click here, now I'm able to do a deep dive and get into the PK data that's relevant for me. So let's start off with some examples to navigate the PK module, and let's start off with trying to answer some pretty straightforward questions first, just to kind of get familiar with the PK module. So for one, we can, for example, ask the question, how can I find protein binding data for the same drug in both animal and human studies? Well, let's take the drug Inablex as an example. Inablex is a prescription medicine used in adults for the treatment of overactive bladder. Let's see what kind of information we can find regarding protein binding in animal and human studies. So how do we search for Inablex? So there's a couple of different ways in which you can do that. One way in which we could do it is we can go to the Add Drugs and Drug Classes tab, and you're going to see a menu pop up on the left-hand side. What we can then do is either search by drug class, or if we are not sure what drug class this drug belongs to, we can go under the A to Z tab. We can click on E and actually search for Inablex over here. Here we can see Inablex, so we click on Add, and we see that it's added over here. Inablex is the brand name, hence we see the original name over here. And now let's narrow the search down to plasma protein binding. So what we can do over here is we see parameter ranges. These are all of the PK parameters that we can search for. And if I click on the Expand button, I can see all the different PK parameters that I can search for right over here. And we have approximately 95 different parameters that you can search for, so it's quite extensive and very useful. So let's go under plasma protein binding. And click the Search Now button. So over here we can see that there's quite a lot of data present for studies done in dogs. And if we scroll down, we can see in humans as well. If we scroll down a bit further, we can see we also get data for mice and rabbits and rats. So there's quite a lot of data that could help you see the view from preclinical to clinical studies. So by looking at this data, we can see that we can gain some interesting insight. For one, if we look at results number 5 and 6, right over here, we can see that by increasing the dose by 5 times, the plasma protein binding does not significantly change in dogs. And that value is seen over here. However, if we look at lines 10 and 11, where we see data from humans, we can see here that by increasing the dose by up to 5 times, the plasma protein binding changes by greater than 90%. And that's seen over here. This can in part explain why adverse events were seen at higher doses. And this is an example of how PharmaPendium can significantly influence the way in which you design your preclinical studies. Here is a nice example of how you can get a nice translational view of what happens in preclinical studies and then how that translates into clinical studies done in humans. So that's one simple example of how we can use the pharmacokinetic module. Now let's try a different example. I'm going to go back to the home page, and now I'm going to show you a different way in which you can search a drug that you're interested in. Let's see if we can find data on a drug called aromycin. Aromycin is a prescription medicine for the treatment of breast cancer. So here, what I can do is type in aromycin in the general search bar and click Go. And now here, because we are looking for all pharmacokinetic data, we will see that there's a pharmacokinetic data tab here, which we can click on. And we will see the view from preclinical data and then clinical data all in one browser. So under the preclinical tab, let's filter and, for example, look at bioavailability data. So what I do is I go under the Show Filters tab, I click on Absorption, and I can select for bioavailability. And I can click on Apply Filter. And right now, I'm in the preclinical data view. So right here, by scrolling down and looking at the data briefly, we can see that, for example, in dogs and rats, the bioavailability is hovering around 5% and under. So now let's see if this data actually translates into humans. All we now have to do is go under the Clinical Data tab and do the same thing that we did before under the Preclinical Data tab. So I'm going to go under Show Filters, click on Absorption, click on Bioavailability, and apply the filter. And over here, we can see that in humans, for the bioavailability data, we see that the value is around 86%. This could be really important data when you're planning your preclinical studies and you have comparative data like this to help you guide your preclinical study design. Now, if you click on the second result, you can see from where they got the data, and in addition, you can find additional interesting information from there. So here's how we can actually click and get into the original document. And here in the first line, you can see where they got their data. And then as you scroll down, you can also see some other interesting data which can help you when you're designing your preclinical studies. Now, let's look at protein binding differences for another drug. I'm going to go back to the Home tab. And because I know the drug name, I'm just going to type it into the search bar. And we're going to look for rosuvastatin. Rosuvastatin is a member of the drug class statins and is used for lowering cholesterol. So let's go under here and see what kind of data we can find. We're going to go under the Pharmacokinetics data tab. Again, similar to what we did before. Now, let's skip the preclinical and go under clinical. And we're going to go under the Show Filters tab. and let's do bioavailability. So here we get a lot of bioavailability data that is present from the clinical studies that were done using rosuvastatin. Now, something that's interesting here is that when you see the drug name, you see the species, you see the doses, whether it's reported or unreported, you see the route of administration, the parameter, and the parameter value, and then the link to the actual document. Now, another thing that you can actually look for is the study group. If I actually scroll down, I can actually see differences between different ethnic groups. We have Caucasian and then we have Japanese. And we can actually see that there are differences in bioavailability between the two groups. Now, as we scroll down, we see that these values are consistent amongst the ethnic groups. Japanese groups have approximately 29% of bioavailability. Caucasian groups have approximately 20%. So this kind of data can really make a big difference if you're looking at, for example, penetrating different markets. This can be something that can be very useful for your strategic decisions. So with that said, what we're going to do now is open it up to a poll, and I'm going to pass it over to Anne-Marie, where she will now conduct a poll, and you can now determine what example you would like to see next.
Yes, Pooja, thank you. So just wait a moment for the poll to appear. And Pooja, of course, has given us some great examples showing us how the PK module works in PharmaPendium. But for the next part of this presentation, we'd like to hear from you and how you would like to see the examples going forward from here. So we have an example. You can vote on seeing an example for metabolite data, looking at an example for Cmax data, or looking at an example for Tmax data. So depending on the time, Pooja might be able to do 1, 2, or 3 of these, but we'd love to hear from you what your order of preference is. So we'll give you a couple of minutes to vote on that, and then we'll close the poll, and Pooja will continue searching in PharmaPendium. So we'll give you another minute to make your choice. And like we said, we will hopefully be able to go through at least 2 of these examples in the rest of the webinar. So great to hear from you what you're most interested in. So let's close the poll and take a look at the results. And we have a clear winner, Pooja. So in top place, 58% of our participants, our audience, would like to see the metabolite data. And if we have time, then the second preference was Cmax data, 38% of our audience. And only 4% would like to see the Tmax data. So certainly the preference is in the first and the second.
Perfect, great, thanks, Amal.
Okay, so we will hide the poll now, and we'll go back into PharmaPendium.
Okay, great. Thanks, everyone, and thanks for voting. So if you want to see how we can find metabolite data using the PK module in PharmaPendium, we again click on the Pharmacokinetic data tab. We can, for example, go under drugs and drug classes, right over here. And as an example, let's go under cholinesterase inhibitors over here. And we can click Add, and you will see all of the drugs which are listed under this class appear over here. We can then expand the PK parameter list and go under biotransformation as an example, and see what percentage of the drug dose is transformed into metabolites. So we click on metabolic transformation right over here, And then we can click on the Search Now tab. And then we can see all of the relevant data pop up. And as you can see, there is a lot of data present, and we actually get data not only in humans, but in dogs and rats. So we get, again, that view from preclinical studies into clinical studies as well. So as we scroll down, what we can see over here, what we can see over here is we have the drug name, we have the species, the study group, the dose, the route of administration. We have the parameter which we are searching for, which is metabolic transformation. We have the parameter value, standard deviation, the time and concomitants if it's described in the study. So if we scroll down, we can see that, for example, the second drug actually has time points over here. Now, this has kind of caught my eye, and so maybe what I would like to do is learn more about it and see what kind of information is present for that study. So what I'm going to do, for example, click over here. Actually, let me click on this one. Here, what we can see, since we saw time points, we can actually see the wealth of data that's present when we're looking at metabolic transformation. And over here, we can adjust the view. But we can see the metabolites. We can see at what time point it was measured, and actually also in what tissue types. So here's an example of why digging into the documents can be really interesting. There's a lot of wealth of data here, which can be really useful for you. Now, if we scroll down a little bit and look at the data provided, we can actually see that there is a metabolite called O-glucuronides. And it's present, we can actually see it's present in both dogs and rats. If I scroll down, I'd like you to just keep your eye out for this metabolite that's listed over here. We can see it in dogs. We don't see it in humans, but we do see it in rats, suggesting that perhaps this metabolite is a species-specific metabolite, which would be interesting to know up front without having to spend too much time wondering if this is something that you need to maybe worry about in humans. So here's just a very simple example of how you can find some interesting metabolite data in the pharmacokinetic module. Now, what about Cmax data? Now, if you want to see how we can find Cmax data using the PK module in PharmaPendium, based on what you have seen already, we can probably not only look for Cmax data, but also ask questions like, is Cmax affected by the fed versus fasted states? Will this in turn affect the bioavailability of the drug? So let's look at an example and see how maybe we can do a deep dive and see how those questions could be answered. So I know of a drug that actually has some interesting data. I'm going to type it in the search bar. So we're going to look at acebutylyl hydrochloride. So again, we'll click on the PK tab. And we can, for example, under clinical data and find some interesting data in humans. So I'm going to click over here. And I'm going to go into the Show Filters tab. I'm going to click on the absorption, and then click on concentration. And then we can actually select for Cmax. And if you're doing more advanced searches, you can actually search for values above or below a certain point. And you can search based on the parameter value that you're interested in. So we'll click on Apply Filter. Now, As a side note, we can actually see that this data is from 1984, and this data would actually be present in the classic collection of PharmaPendium, which has data from 1938 till 1991 inclusive. This could be interesting info if, for example, you're looking at PK data for repositioning this drug on the market. That's just a little aside. So from looking at this data, we can actually see that by doubling the dose, we can almost double the Cmax value. So here we see 200 milligrams, and then over here we see 400 milligrams, and we have the Cmax values present here, which are almost double in value. Now, if we click on, let's say, the third result, because it actually also shows us data on the metabolite, which is listed here, and on acid state, we can actually see that it gives us further interesting information. I'm going to scroll down a little bit. So it actually says that the study demonstrated that although non-fasting conditions resulted in a decrease in the rate and the peak levels of acylbutylol absorbed and metabolized by these healthy volunteers, there was no significant difference in the actual bioavailability. And here's a table which shows that info. So obviously, by doing a deep dive into some of the documents, you get some very interesting comparative data here for drug and development study. Now, if you did not have access to the pharmacokinetic modules, you would not be able to click on the link and do a deep dive into the original documents. So here's an example of how really clicking on those links and getting access to that original document, which refers back to the extracted data, can be extremely useful. So since we have a couple of minutes left, I actually think we have time to do the last example, which is in relation to Tmax data. So now, if you want to find some interesting Tmax data, which is data pertaining to how long it takes to reach Cmax, we can, for example, look at analgesics. So let me go back and select analgesics. And then under the PK parameters, let's go back. Here are all the analgesics listed, quite a lot of them. We can click on distribution, click on time values, and we'll get the Tmax. We'll go back up on the Search Now button. And here you can see all of the results. And as I scroll down, you can really see all of the data that's available. In some cases, you can even see age groups, which can provide some very interesting data. Over here, you have the year in which the study was performed. So there's a lot of really interesting data that's present over here. Now, it is pretty straightforward to see all of the data that is available. But let's see if we can find an example of some interesting data we can find with combinatorial drugs. So if we scroll down, we can see data on combinatorial administration of acetaminophen, aspirin, and caffeine. So let's scroll down and find that. So here, for example, we see the combination of acetaminophen, aspirin, and caffeine. Here we see the doses listed in the order that the drugs are listed here. So let's click on the link to go straight to the document, see what kind of data is present over here. So the document actually puts data in comparison to different forms of the drug. So they have it actually in the form of a tablet, caplet, and gel tabs. So that is very interesting data that you can have right there and then. And you can actually see, after scrolling through all of the data, for example, this last table shows you data, summary of acetaminophen pharmacokinetic parameters after administration of Excedrin extra strength formulation. And so there's a lot of data here that is present in the tables. And so you can actually see at the end of scrolling through all this data that the concentrations of the combinatorial drugs is not affected by the extra strength acetaminophen, which is estrogen. This data can be really useful if, for example, you're doing similar combinatorial drugs. This data can potentially be cited and could maybe help you avoid repeating studies, which can save you a lot of time and money. Now let's just go back and take a different example in this search. Again, we'll try to search for a combinatorial drug. Let's maybe go down here. So we have here acetaminophen and tramadol hydrochloride. So if we click on the link and scroll down to the Pharmacokinetic section, over here, we have the Pharmacokinetic section. We have more data that is available to us by doing a deep dive into the original document. We can actually kind of quickly look at the paragraph that's under this document. So here I'm just going to read some of the information that's present here, which can be really interesting. And we can actually see that there's a difference in PK profiles of tramadol, not acetaminophen, when administered together. So here it actually says, upon multiple oral dosing to steady state, however, bioavailability of tramadol and metabolite M1 was lower for the combination tablets compared to tramadol administered alone. And then if we look at the last line, it says, following single or multiple dose administration of alpha-set, no significant change in acetaminophen pharmacokinetics was observed when compared to acetaminophen given alone. So here's an example of something to keep in mind. This data, where we see one drug being affected by acetaminophen, is different from the previous example I showed, where acetaminophen did not significantly affect the PK profiles of the other combinatorial drugs. Hence, being fully informed on the comparative data which can be available to you is critical in order to properly plan out your preclinical and clinical studies. So those are some examples of how we can use the pharmacokinetic module. I will then pass on to Anne-Marie, who will then open it up to Q&A.
Yes, thanks, Pooja. Fantastic examples. Thank you for taking us through that. We have a couple of questions. And one was from a user of PharmaPendium who's wondering why they cannot see the pharmacokinetic button that you're pressing or that you're clicking on. Perhaps you could give some explanation or some details as to how they can see that pharmacokinetic button.
Oh, sure. I'm sorry. I probably should have mentioned it at the beginning. So the pharmacokinetic module is something that needs to be purchased in addition to the PharmaPendium platform. So if you have not purchased the pharmacokinetic module, you will not be able to see the tab that's over here, and therefore you'll not be able to do a deep dive into the preclinical data that's present in PharmaPendium. So the PK module needs to be purchased in addition to the PharmaPendium platform.
Okay, thanks, Pooja. We have another person who would like to find out a little bit more information about telling the differences between drug classes. How do you go about finding information that would tell them the differences between drug classes?
So why don't we go into the pharmacokinetic data tab. Here what I'll do is, again, I'll click on the Add Drugs and Drug Classes link. I'll get all of the drug classes here, so you can choose whichever two drug classes, for example, that you would like to compare. So maybe for this example, what we could do is look at adrenergic agonists. I can add that there. And either I can add another drug class, or if you want to look at two drug classes separately, you can actually do two different searches. So what I'll do is I'll first do the search for the adrenergic agonist, and then I can scroll down for whatever PK parameter that is of interest to me. So why don't I do the expanded view. And let's look at AUC values. And I'll click on the Search Now tab. And you can see here are the search values listed under the parameter value. And by scrolling down, I can see that I can get an idea of the range of values that are present, for example, in that drug class. Here I can see a range of picograms to nanograms. And this might be a characteristic of that particular drug class. In a side-by-side tab, what I could do is conduct the same search but with a different drug class. So maybe go back to what we looked at before, analgesics. And I select for AUC like I did last time. Scroll back up, click on Search Now. And then here I can look at the values and look at the range. And here I see that a lot of the values are in the micrograms, potentially down to the nanogram values. So here I have a side-by-side view, so I can do a direct comparison between the two. So in a nutshell, we see that for adrenergic agonists, AUC values range picograms to nanogram values, and for the analgesics ranges from the nanogram to microgram values. So that's a way in which you can compare drug classes, and it's very easy by opening up, let's say, two different tabs. You can do a side-by-side comparison, or if I were to do them both at the same time, I can just go under here, and I can add adrenergic agonists, and I can also add analgesics. And I'll see everything added here, and then I can conduct the search that way. So hopefully that answers the question.
Thanks, Pooja. And just while you're, we're almost out of time, as you're bringing up the last slide, I would like to say that we do have a couple of questions that we didn't get to today, so apologies for that. Such as we had one question on collecting repeating data into one line if it's originated from the same study. We had a question on the cost of PharmaPendium, and these are all questions that we will get to straight after the webinar and fill them in and send to all of you who were able to join today. So you will have a copy of those questions and answers from the webinar in your inbox. And I would like to say thank you once again for attending today's webinar. Thank you to Pooja for the excellent information and for the examples. If you have any questions or comments on the information you saw today or any training material or information around PharmaPendium, then you can send an email to bdtraining@elsevier.com and we will get back to you. Today's session offered an overview with examples of searching for PK data in PharmaPendium, and as I said at the beginning, we also offer introductory sessions, and the next one will be on June 27th. So if you or your colleagues are interested in an introductory session, please do join us, and we will send you the link to the webinar calendar in a follow-up email, and please feel free to register for as many PharmaPendium sessions as possible, or as I said, send it through to your colleagues. And please do keep in mind that we also have a PharmaPendium newsletter, which we will also include, and this is the best way to make sure that you stay up to date with what is happening in PharmaPendium and in the industry. We often include case studies and interesting search tips and articles to download as well, relevant to the area that you're working in. When you leave the session, a survey will pop up. Please fill out what your thoughts are regarding this webinar. This will be really useful for us to improve the webinars going forward. And we will send a link to this recording and send you the slides as soon as possible after this session. Many thanks again. Good luck with further exploring PharmaPendium. Don't forget to fill in the survey. We'd love to hear from you. And thanks to Pooja, and we hope to meet you again soon.